Oh, my appointment. No. All right, you guys can hear me on. I guess nobody's gonna give me a thumbs up.
Yeah.
Okay.
Yeah.
Yeah, no, this one's stuck. It might be stuck. That's all right.
That's okay.
We just start with that. Yeah, sure. Yeah.
So you can move.
Testing, testing. Cool.
All right. Hello, everybody. I know kind of a different face here today. So my name is Clayton Ivey. I'm a fourth-year undergraduate in Dr. Jacqueline Lindbergh's lab. Currently studying nutrition, exercise, physiology. So this past summer, I actually got the opportunity to work with our guest today. So it's really, really cool that I get the chance to kind of come up here and introduce him. So yeah, so I actually met Dr. Robinson in 2019 at the annual biomedical research conference for minority students. I was there presenting, and this guy kind of comes up to my poster, and he's like, "Jackie Lindbergh?" I'm like, "Yeah, like what's it to you, man?" So we got to talking a little bit. I got to present my work to him, and I actually ended up winning an outstanding poster presentation award, which was pretty cool. And so later on, I was looking for, an opportunity for a lab outside of Dr. Lindbergh's, because I had spent a couple summers there already. And Dr. Robinson, I reached out, and he was graciously just had me down and gave me the opportunity to come and learn. So it was awesome. Just a great experience. So I'll go ahead and get talking about him. So Dr. Robinson, he's originally from South Bend, Indiana. He actually graduated undergrad with a bachelor's of science in exercise science at the University of Indianapolis back in 2010. He then moved on, did his master's and PhD at the University of Illinois at Chicago, working in the lab with Dr. Shane Phillips. And so, yeah, during that time, he also spent time as a teaching and research assistant, as well as an adjunct lecturer at Benedictine University. So he kind of got to do everything. Pretty cool. So he then spent time as a postdoctoral researcher in the lab of Dr. William Farquhar at the University of Delaware. So obviously pretty big guy there too. So before securing, or that's what he did kind of before securing his current position as an assistant professor and the director of the neurovascular physiology lab at Auburn University. So he's a member of the American Autonomic Society, American Physiological Society, American College of Sports Medicine, American Heart Association, Microcirculatory Society, and the National Strength and Conditioning Association. He's received numerous awards, including the Stephen M. Horvath Professional Opportunity Award from the American Physiological Society and the Certificate of Research Excellence from the American Heart Association. Dr. Robinson currently has 37 publications. He's just a beast in the field, with a few others in review. So he's been continuously funded by the National Institute of Health since 2012 at UIC, and he currently holds a 5-year NIH KO1 award to study racial differences in serum sodium and blood pressure regulation. So today he's going to give a talk about the influence of dietary salt loading and race on cardiorenal function in healthy young adults. So enough of me, Dr. Austin Robinson.
So thank you for that kind introduction, Clayton, and thank you all for being here today. So as Clayton mentioned, the talk is the influence of dietary salt loading and race on cardiorenal function in healthy adults. So briefly, I'll just go through my background kind of as a trainee, personal background. Clayton touched on that already a little bit. My interest in salt and blood pressure independent effects of dietary sodium. So we'll specifically focus on kidney function and endothelial function. We'll talk about some of the current work that I'm doing with racial differences on the effects of sodium. Finish up with some future directions, so some work that we have in progress related to salt and sympathetic nerve activity, and then questions and answers. Okay, so as Clayton mentioned, I'm from Northwest Indiana, did my bachelor's degree at the University of Indianapolis. And then I moved over to the University of Illinois at Chicago, where I did my master's degree and PhD with Shane Phillips. In 2016, moved out to Delaware to work with Bill Farquhar, did a 3-year postdoc, and then in 2019, started a lab at Auburn. And so I've been there ever since. In terms of my research interests, I am interested in cardiovascular disease or heart disease. Pointer work? Guess not. So that's the leading cause of death for both females and males in the United States. And then kind of closely behind, you can see that stroke is also a leading cause of death for both females and males. So together, cerebrovascular and cardiovascular disease are a major cause of death and morbidity. And then from a financial perspective, stroke and cardiovascular disease cost about $350 billion per year. So that includes things like medical bills, medication costs, time lost from work, kind of these indirect and direct costs associated with these diseases. And so just to kind of put things in perspective, so as of last week, there was a little over 700,000 deaths attributable to COVID-19. So that's obviously terrible. We all know a lot of people who've either had COVID or maybe kind of directly or indirectly people who have passed away from COVID. But again, a little over 700,000 deaths through November 9th. COVID came to the United States in March 2020. So that's about 1.5 years, a little bit longer. And then the most recent data from cardiovascular related deaths from 2019, about 660,000 deaths, and this is year after year after year, not this, a temporary pandemic. So just to kind of put things in perspective, cardiovascular disease is definitely a major cause of mortality. And so one of the leading risk factors for cardiovascular disease is hypertension. So what we can see on this graph on the x-axis is age, and then on the y-axis, we're looking at percent of the population. And so we know that there's this age-related increase in hypertension. And then one thing to point out is this is data from 2013. So in 2017, the American Heart Association and American College of Cardiology kind of reclassified their definitions for hypertension, making it more stringent. So for example, to have hypertension, you used to have a systolic blood pressure of 140 or above. They lowered that to 130 or above. So same trends, but if anything, now we would see higher percentages across the age span. Okay, so next I'll kind of transition into dietary sodium. So a little meme here, threw sodium chloride at me, that's a salt. This is data from the New England Journal of Medicine in the PURE cohort, so over 100,000 adults across different continents. And so what we're looking at on the x-axis is urinary sodium excretion. So it's not perfect, but our kidneys do a pretty good job of trying to excrete excess sodium. So if you eat more sodium, you're going to urinate more sodium out to try to get rid of that excess sodium. So we can see as urinary sodium excretion increases, we also see an increase in systolic blood pressure. And this is something that most of us are familiar with. Salt, yeah, it's associated with blood pressure, but this is at the population level. So there's a lot of variability between individuals as to whose blood pressure would increase with salt and whose blood pressure would not increase with salt or decrease if they reduce sodium. And then this data was also, there's this relationship kind of remained even after adjustment for important covariates like age, sex, BMI, alcohol, and geographical location. In terms of dietary sodium consumption in the United States, the recommendation through the dietary guidelines for Americans is 2,300 milligrams per day. And the American Heart Association recommends 1,500 milligrams per day, especially if you have hypertension or if you're trying to shoot for optimal cardiovascular health. And then the average American consumption is 3,500 milligrams per day. So that varies by sex. So males on average consume closer to about 4,000 milligrams per day, and females tend to consume closer to about 2,900 or 3,000 milligrams per day. And so we know that excess dietary sodium, so consuming around 4,000 milligrams per day, is associated with the 17% increased risk for cardiovascular events. And then in terms of sodium and what we know about sodium and hypertension, there are some pretty cool ethnographic studies. So I'm going to show some data from the Yanomami Society, and they were compared to another neighboring tribe. So these are both kind of societies on the border of Brazil and Venezuela. And so with the Yanomami Society, they consume a really low amount of sodium. So if we did like a controlled feeding study, maybe the low sodium group would receive 1,000 milligrams per day. They habitually consume less than that. And there's other things going on, of course, like they probably have more physical activity than the standard American. They probably have less distraction from things like blue light and screens and things like that as well. But what's interesting is that they were compared the Yanomami to the Yakwana, who have taken on kind of a more Western style diet because they engage in more trade, so they consume more processed foods. And so what we can see from their data is that on the left, in the upper left here, in the Yakwana individuals, that there's that age-related increase in blood pressure, kind of similar to what we would see in America, maybe to a little bit lesser extent. And then in the Yakwana Society, across the age span, so as young as infancy all the way up to about 60 years old, we can see there's a pretty flat line in terms of systolic blood pressure. And then we see similar trends for diastolic blood pressure as well. So that would suggest that at least in this society where they have very low sodium consumption, there's not that kind of what we perceive to be inevitable age-related increase in blood pressure. All right, so in terms of blood pressure independent effects of sodium, this is data from the trials of hypertension. Can you still hear me? Okay. So from the trials of hypertension prevention, so there's trials 1 and 2. And so they looked at cumulative incidence of cardiovascular events, so things like myocardial infarction, cardiac arrest, stroke. And what they found is that in TOHP1 over follow-up, there was less cardiovascular events in the sodium intervention group. So during the actual study, the individuals were counseled to consume less sodium. So they changed things like the type of bread they ate. And then they kind of had foods prepared for them to kind of coach them and counsel them how to reduce their sodium. And then throughout follow-up, while they didn't reduce their sodium consumption to the same extent as when they were actively in the study, in that post-surveillance period, they still maintained lower sodium than the individuals that were initially in the control group. And so we see that reduction in the cardiovascular events, despite in these two groups, there being a pretty modest difference in blood pressure over time. Okay, so in terms of the foods that are contributing to our sodium intake, the American Heart Association has this list of the salty 6. So these 6 foods make up about 42% of the average American's sodium content in their diet. So one, we have breads. So if you go to the store and you buy like prepared bread or loaves or things like that, that usually has sodium in it already. So that's something you can't really control. Poultry, so sodium is added to poultry. So if you add sodium, that's going to cause more water retention. So then if you go buy that meat, that's going to weigh more and they can charge more for that. So that's one of the reasons why sodium is also included in poultry and meat products usually. With sandwiches, that's kind of a multi-ingredient food. So it's got both the cold cuts and the bread. Same thing with pizza. So it's usually got like pepperoni or some sort of processed meat and bread. Condensed soup is also a major source. And then, as I mentioned earlier, cold cut and cured meats. So just for an example of some common foods that most of us wouldn't even think were that outrageous in their sodium content, if we look at Papa John's on the far left, so I just pulled data for 3 slices of pepperoni pizza, that would be 2,700 milligrams. So that surpasses the DGA guideline for 2,300. If you got an Italian BMT at Subway, you can see that that's about 1,100 milligrams. So that gets you close to halfway. A can of Campbell's Chunky Chicken Noodle Soup has 1,700 milligrams. So there's 2 servings in that can, but I mean, I don't know about you all, but I don't think most people eat 1 serving of the soup. They usually just pour the whole can. And then with the Chipotle steak burrito, that's about 2,300 milligrams. So it puts you right at the guidelines. So in terms of where sodium is coming from, there's data from 2017 where they pulled data from California, but I think it was UCLA was the site, Minnesota, and then UAB, so Birmingham, Alabama. So across 3 different sites in the United States. And what they found was that processed food in the dark blue here makes up about 70% of our dietary sodium intake. And then if you think about like what's added at the table, it's only about 6%. So when I did a lot of the sodium work I did at Delaware, we'd have people come in and they'd say, oh, I don't really add that much salt to my food, so I don't think I eat that much. But you can see adding salt at the table isn't really a huge contributor for most people. And in terms of what we'll focus on today, outside of kind of where sodium is coming from, is the effects of salt on vasculature, and then also with the kidney and renal dysfunction. Okay, so we'll start with the kidneys. So we know that dietary salt is associated with future CKD from a number of epidemiological studies. And there's a number of potential mechanisms. So one could be increased aortic stiffness, leading to increased pulsatility in terms of blood flow to the kidney. And there's also a growing interest in biomarkers that you could use to potentially screen for those who are more at risk at having kidney damage if they consume high sodium. And so there's a number of biomarkers that you could use. The two that we'll focus on are neutrophil gelatinase associated lipocalin, or NGAL, or kidney injury marker 1, which is KIM1. And so in terms of the utility of these two biomarkers, we know that NGAL has been associated with CKD progression and severity, and then also in the ERIC study, it's been associated with CKD stage 3, and then KIM1 was associated with incident CKD in another large cohort study called MESA, or the multi-ethnic study of atherosclerosis. Right, so in the lab, what we did was we had 24-hour urine collections. So people would come in under these different conditions and they would collect urine for 24 hours. And then we would check that they collected it for an adequate amount of time, and we can calculate their urine excretion and then normalize that to the time that they used the container and calculate what's called urine flow rate. So basically the volume in milliliters divided by the time in minutes. So that'll be important for interpreting one of the graphs in a few slides. And so we did our basic measures of kidney function. So serum creatinine, we can also measure urine creatinine, and between those two calculate creatinine clearance, which is a measure of glomerular filtration rate. And then as a backup, we also measured cystatin C. So there's some controversy with urine creatinine that can be influenced kind of by proximal tubule release of creatinine. So if you measure cystatin C, it's thought to be released at kind of a constant rate from all the cells in our body. So you can measure that. And then we also measured urea. And then the two biomarkers, like I said, were NGAL and KIM1. And then we measured those using ELISAs. So in terms of what we knew from other data that preceded ours, so there's some preclinical data in rodents. So they were given different doses of sodium chloride. And so what we can see in this graph up top here, 8% sodium chloride group, within about 1 week, there's an increase in urinary NGAL excretion, even when it's normalized to creatinine. And so this is in male Wistar Kyoto rodent or rats. And then there's also some data in spontaneously hypertensive rats as well. And then the thing that was interesting is that there was no change in serum creatinine or urine albumin, so no proteinuria. So that change in NGAL was kind of more sensitive compared to some of the more common measures of renal function that we're used to seeing. And then last year, Kristen Nowak and Doug Seal's group published some data in older adults. So this is data from a study where older adults either consume their normal sodium diet, so habitual diet, or they were counseled to reduce their sodium for 5 weeks. And so it was a relatively modest sample size, but it was a good study in the sense that they had them do these two interventions for 5 weeks. And so on average, urinary sodium excretion was reduced from 150 millimolar per day to about 66. So just for some context, 150 millimolar is pretty close to like the standard American diet, 3,500 milligrams. And you can see that they cut that by more than half. So pretty drastic reduction. And there was a 31% reduction in NGAL. So you can see that in the top bar graph with the group average data. And then below that on the right, we can see the individual data. So most of the participants out of the 13 demonstrated a reduction in urinary NGAL. Okay. And they also tried to assess KIM1 in this study, but it was immeasurable in most of the samples. Okay, so for our study, it was in healthy young adults. So we did screening, we performed a VO2 peak, and then we had participants consume either dextrose, so that was our placebo, or sodium capsules, which was sodium chloride, for 10 days each. There was a 2-week washout, and then they crossed over into the other intervention, whichever they didn't start out with. And so in our cohort, it was recreationally, healthy young adults. So also a little different from the prior study in that they used older adults. So these are our participant characteristics. You can see on average, they're 24 years old, normal BMI, or VO2 was about 40. So again, they were recreationally active, not like elite athletes or anything like that. And on average, they had a healthy blood pressure, so 111/64. And this data was led by my recently graduated master's student, Alex Barnett, who Clayton got the chance to work with. So when we looked at plasma NGAL, there was no difference, which this was kind of to be expected for us. We wouldn't expect there to be a change in plasma NGAL unless there was some pretty serious kidney damage. This was just a 10-day high sodium load. But what we did see was that there was an increase in urinary NGAL concentration. And then when we normalized NGAL to urine flow rate, because when you consume more sodium, that often changes how much you drink and how much you urinate. So it's important to index to flow rate. And what we found was that there was still a significant increase in the urine NGAL. And then when we indexed to creatinine, we found there was no change, which we did find that to be interesting. And I'll explain why that is in just a couple of slides. And so with the KIM-1 data, we found no difference in plasma concentration or when we indexed the urine KIM-1 to flow rate, or if we indexed it to creatinine. And so this is where we found this data to be kind of interesting because again, this is a healthy young cohort and we found that there was a significant increase in urine creatinine. So in an older cohort, or if we had a cohort with patients with hypertension, we might expect the sodium to change creatinine clearance, but we didn't necessarily expect that in younger adults. So the literature in that is kind of mixed. In most studies with younger adults, you don't see a change in creatinine clearance, but there are a couple prior studies that have shown that. And so when we calculated creatinine clearance, it was significantly elevated. If we looked at the data categorically, just looking at high GFRs, so above 135, there's also a significant difference. And then when we adjusted for body surface area, there was still an increase in creatinine clearance, but not when we looked at the data categorically. And I think that was just because of our small sample size. And then urinary cystatin C was also significantly increased. So that gave us a little more confidence in terms of that urinary creatinine clearance measure. And then there was no difference in urinary urea excretion. And then as you would expect, there's an increase in fractional excretion of sodium just because they're consuming more sodium. And so the reason we kind of found that interesting as well is because hyperfiltration, so what we were witnessing in our cohort, is also associated with future cardiovascular events. So compared to normal GFR, both low filtration and hyperfiltration are linked with future cardiovascular events. And with the hyperfiltration, that could be causing damage to the microcirculation within the kidney and could contribute to future declines in GFR over time. So to summarize our renal data, so we know that high salt intake is associated with CKD over time. We found that even in a cohort of healthy young adults who are on average only about 24 years old, that high salt increased GFR. It also increased NGAL. So that would suggest that if these participants did consume a high sodium diet over time, that could be one link to why they might be at increased risk for CKD. So next we'll pivot and talk about some of the data on dietary sodium and endothelial function. And so just kind of a brief overview of endothelial function. So there's three layers here: the tunica intima, which is the innermost lining of our blood vessel, that's the endothelium. That middle layer is the muscular layer, or the tunica media, and then the outermost layer, the tunica adventitia. So with endothelial function, we normally assess how functional the endothelium is by looking at different pharmacological agonists, or we could look at increases in shear stress and nitric oxide and vasodilation. So normally an increase in shear stress would cause the endothelium to release nitric oxide, and then that vascular smooth muscle layer to relax or vasodilate, and the blood vessel would expand or get larger. And so first we'll talk about one of the techniques that we use to assess large artery or conduit artery function. So that technique is flow-mediated dilation of the brachial artery. So in this case, our model is Clayton there. And so one of the grad students, you can see, has a probe in their hand, and they're using that to image his brachial artery. And then just distal to his elbow, there's an occlusion cuff that we can rapidly inflate and deflate. So after we've captured 1 minute video of both blood flow and artery diameter, we would inflate that cuff to a suprasystolic pressure. So in our lab, we typically use 225 millimeters of mercury for 5 minutes to occlude blood flow, and then we would release that pressure in the cuff and then continue to capture blood flow and blood vessel diameter for an additional 3 minutes. And so the reason we care about this measure is it's been associated with cardiovascular risk prospectively. So changes in that are negative are in theory, should be a bad thing. So this is what we would see in our lab if we kind of pulled that image into the computer, so we could track blood vessel diameter and then blood flow velocity. And from those two variables, we can calculate blood flow and shear rate. And then this would be kind of representative of post-occlusion period. So you can appreciate that there's this large increase in blood flow velocity. So there's this increase in blood flow. And that increase in blood flow, again, should cause the release of nitric oxide. And there's other vasodilatory factors, but it should cause the blood vessel to dilate in a normal, healthy condition. And so what we know about dietary sodium and endothelial function, so a lot of the data I'll present here was done by students and professors at the University of Delaware, even before I started my postdoc there, kind of led by Dave Edwards and Bill Farquhar. And so for their initial study where they did some of this work, they had a run-in diet with medium sodium or recommended sodium for 7 days, and then they were randomized to consume either high sodium or a very low sodium diet for 7 days each. So for the high sodium diet, it was close to 7,000 milligrams per day. And then for the low sodium diet, it was 500. So what we can see here is that the intervention was effective. So you can see that there's this large increase in urinary sodium excretion, so close to 300 millimolars per day, which is about where they wanted it. And then with the low sodium diet, you can see it's less than 40 millimoles per day. And so in this cohort, they were what we would call salt resistant. So when we look at the bottom left, the 24-hour mean arterial pressure, those two bars are pretty similar. So there wasn't a very large change in mean arterial pressure. So we would classify this cohort as salt resistant. And then if they were what we would call salt sensitive, you would see an increase on that white bar. So there would be an increase in pressure. So just to kind of clarify, this is a continuum. So if you measure hundreds of people, you're going to have some people experience a pretty modest change and it's going to go up. But usually for the sake of studies to kind of put people in these different bins, you'll have that 5 millimeters of mercury kind of cut off for mean arterial pressure. So if they're less than 5, usually would classify them as salt resistant. And if they experience a change in mean arterial pressure of more than 5, you'd classify them as, if they're less than 5 salt resistant, more than 5 salt sensitive. So despite this cohort being salt resistant, they still found a significant reduction in endothelium-dependent dilation, so the brachial flow-mediated dilation. So you can see the individual data up top. Most of the participants did experience that reduction in brachial artery flow-mediated dilation. And then if you look at the group average data, you can see on average there was a reduction. So next, I'll discuss a technique that's used to assess small artery function or resistance artery function. And so this was a technique that I picked up during my PhD in Shane Phillips' lab. And so this is pressure myography. So we would obtain biopsies, usually adipose biopsies from subcutaneous abdominal adipose or gluteal adipose tissue. And then we would dissect small arteries from that adipose tissue under a microscope. You have to be really careful not to damage them. So you use these fine tip forceps. And so once you've cleaned the artery up, you can cannulate it on 2 glass micropipettes. So in this organ chamber here, you can see in the baby blue, that's just kind of a representative of the glass micropipettes, which you can see kind of in closer detail on the right. So you can tie the artery in place using these fine nylon filaments onto the micropipettes, and then they're pressurized with the physiological saline solution or Krebs buffer. And then there's also provided gas and kept at about 37 degrees Celsius. And that Krebs buffer is kind of a rough mimetic of blood. So it's got electrolytes, it's got glucose and things like that to keep the vessel alive and healthy. And so in terms of assessing endothelial function, you can pre-constrict these arteries with things like endothelin-1, and then use acetylcholine or different agonists to cause endothelium-dependent vasodilation, or you can change the height of these reservoirs to create a pressure gradient, so mimicking an increase in blood flow to cause endothelium-dependent dilation as well. So this was a project that was going on in our lab by two postdocs at the time where they were interested in dietary salt. And so what we can see here on the x-axis is the pressure gradient. So again, that pressure gradient that you can create by changing the height of those reservoirs. And so as you increase that gradient, you can see that there's this increase in flow-induced dilation across those gradients. And then if they add a compound called L-name, which inhibits the enzyme nitric oxide synthase, so that's basically inhibiting production of nitric oxide, we can see that under normal conditions, there's a significant reduction in endothelium-dependent dilation. So this is under just normal habitual conditions, and then participants received sodium capsules to increase their dietary sodium. And so we can see that there's both a significant reduction in endothelium-dependent dilation and then less of an effect with the L-name, suggesting that there is a reduction in the nitric oxide component to vasodilation or NO bioavailability. Okay. So just to kind of recap, this is again that same study that we used, the salt capsule study in healthy young adults, and we were also interested in determining if we could use that model to also investigate changes in endothelial function in contrast to the prior controlled feeding study, and we're interested in blood pressure responses to dynamic exercise. So this is back in the postdoc lab when I had much shorter hair. You can see my lab mates here testing my blood pressure during dynamic exercise. And so we're interested in the blood pressure response to exercise because blood pressure responses to different stressors are prognostic of future hypertension. So what we're looking at, so the data on the right is from a study, unfortunately, is in all males, but they brought them in for an exercise test and they quantified the change in blood pressure during the exercise test. And so they broke it into quartiles. And so the first quartile, so that bottom group with the hashed lines, were those who had the lowest 25% of a blood pressure response to exercise. And then in the top group, in that solid line, the fourth quartile, that would represent individuals who had the largest blood pressure response to an acute bout of exercise. And so what we can see is over time, the group that had the largest blood pressure response to that physical stressor was also much more likely to develop hypertension. So your blood pressure response to a physical stressor could indicate some level of blood pressure dysregulation that's occurring early on. So in our study, we found that there was a significant increase in systolic blood pressure during a bout of cycling exercise. So just to kind of orient you here, we did a 5-minute warm up and then 50 minutes of exercise at 60% of the intensity from a maximal VO2 peak test, and then a 5-minute cool down as well. So there was a significant increase in systolic blood pressure. And then if we just look at the average change in systolic pressure, that was also elevated. There was no change in mean arterial pressure or no change in the diastolic pressure. And so we were kind of curious as to why specifically systolic. So we did correlate that to changes in plasma volume, which we can estimate based on changes in hemoglobin and hematocrit. And we did find that there was a correlation between the increase in systolic pressure and the plasma volume expansion. And as I said earlier, so we're also kind of interested in maybe determining if you can use that salt capsule model to also study endothelial function rather than just controlled feeding. And similar to the controlled feeding, we did see a significant reduction in flow-mediated dilation. And it was interesting, there was some previous group had shown a correlation between FMD and systolic blood pressure responses. So we wanted to see if that also applied in the context of this study. So what we're looking at here is change in FMD. So the further you go to the left, that's greater percentage change in FMD. And then on the y-axis, we're looking at change in BP. So those who had the largest reduction in endothelial function also tended to have the greatest increase in systolic blood pressure on the high sodium compared to habitual sodium condition. So if you're interested in salt and endothelial function, there's about 20 papers that have been published now on human participants where they've given sodium and measured some index of endothelial function. So Jordan Paddock, who's a current postdoc at Delaware, published this paper last year, and it's really good summary of those papers and some of the mechanisms that have been proposed. So one of those is that sodium increases NADPH oxidase expression. So that's a pro-oxidant enzyme that produces superoxide, and superoxide can scavenge nitric oxide and reduce endothelial function. And then something we won't talk about too much in this particular talk, but sodium also suppresses the renin-angiotensin-aldosterone system. And so angiotensin 2 is a transcription factor for superoxide dismutase, which is an antioxidant enzyme. So by suppressing Ang2, you can also decrease SOD activity, and that's kind of less endogenous antioxidant defense that you could have. And then there's also some data, mostly in cell models, looking at the influence of sodium on the glycocalyx. So that's basically like the mechanotransduction kind of pathway of how you can sense that change in shear stress and lead to vasodilation. So by disrupting the glycocalyx, that also impairs endothelial function. Okay, so next we'll talk about racial differences on the effects of sodium. And so that's what I'm focused on for my K award. And so I talked about salt sensitivity and how there's kind of variability amongst individuals in terms of some people can eat more sodium and experience no change in blood pressure. Some people, if they reduce sodium, their blood pressure will go down, or if they consume more sodium, their blood pressure would go up. And so one group that's been identified as being particularly salt sensitive is African Americans or Black Americans. So if you took a group of 100 Black Americans compared to 100 white Americans and put them on a high salt diet, you would see that in the Black Americans, there'd be more salt sensitivity or a higher proportion of salt sensitivity. And so this is data that I helped pull together and write up as a postdoc. So this is from a hypertonic saline infusion study. So you place an intravenous catheter and then you can infuse saline. So if you infuse normal saline, that shouldn't really have an influence on serum sodium concentration, or you can infuse hypertonic saline. So saline that has a higher sodium concentration than our blood. And so that's going to increase the sodium concentration in our blood. And that was what was performed here. So it was a 1 hour infusion of a hypertonic saline. And we can see to start out with in the black filled circle that the African American group, if anything, had a slightly lower serum sodium compared to the Caucasian group. And this is in healthy adults on average. They're in their mid-20s. And over the course of that hypertonic saline infusion, we can see again on average that the African American cohort ends up with a slightly higher serum sodium. And then for any given serum sodium, we can see that on average, the Black cohort had a higher blood pressure as well. So when we looked at the data categorically, so a modest change would be a serum sodium change of about 0 to 3 millimolar. Moderate would be 3 to 5. And then if your serum sodium changes more than 5 millimole, that's a pretty large change because the kind of operating range is like 135 to 145. But across all these different kind of changes, we saw that the African American group had a larger change in systolic blood pressure. So there seemed to be kind of more of a sensitivity to the changes in serum sodium. And then we're also interested in the ability to buffer those changes in serum sodium. And so what we found was that about 51% of the African American cohort experienced a large change in serum sodium compared to just 1/3 of the Caucasian American group. And so when we looked at that, statistically, it wasn't quite significant. P equals 0.06, but this is in just 39 adults. So it's probably just kind of underpowered, but we think there was a pretty decent effect size there. So one of the limitations to this study is, again, using that hypertonic saline infusion. It doesn't necessarily translate to the real world. So we wanted to use a more physiological model. And so that's what we're doing now. We're going to use, or we are currently using, a soup study. So we're giving participants a high salt soup and a low salt soup. So a lot of the prior studies that have done these acute soup feeding studies to look at dietary sodium and the acute effects have used 1,500 milligrams in a meal. And so we're using 2,500 milligrams just because, as we pointed out earlier, it's not uncommon for Americans to consume more than the dietary guidelines in one sitting. And those weren't even like outrageous meals. So if you went out to like a fast casual place and got an entree and appetizers and stuff like that, you would consume like north of 3,000 milligrams in one sitting. So we're using 2,500 milligrams in this study. And then in a prior study that we did at Delaware, we did find that there were significant changes in serum sodium with just 1,500 milligrams of soup or sodium in the soup. And in this study, there was kind of we weren't interested in racial differences per se, and we're actually more interested in the cerebrovasculature here. But it was some pilot data to kind of decide, yes, we can elevate serum sodium with just one meal.
And within 60 minutes, right? That's pretty fast, isn't it?
Yeah.
How fast would that go down? How fast does sodium clear?
And some of the studies that have tracked it a little longer, it usually starts to come down by 2 or 3 hours. So one of the things that we want to dispel with this study is this slavery hypertension hypothesis. So this has been around since like the late 70s, early 80s. And there's really kind of three tenets to this hypothesis, but it really picked up steam, I guess just because it comes across as biologically plausible. So the thought was that during the transatlantic slave trade, just due to lack of water, lack of food, individuals who could retain more sodium would be better off. So the thought was that slavery created this artificial selection pressure for sodium retention, and that could contribute to salt sensitivity. And there was a 2007 or 2008 episode of Oprah where Dr. Oz was on, and they talked about it. So then, it's even kind of pervaded into like pop culture and just like the lay population. A lot of people think this. I remember I had a history professor in high school who taught us that, and I was like, oh yeah, that makes sense. So anyways, it's called the African gene hypothesis or slavery hypertension hypothesis. And so there's kind of three speculations here. One is that there's, the hot and arid environment of West Africa that could have contributed to some selection. Then the transatlantic slave trade, and then the actual slavery that occurred throughout like the southeastern United States. Because again, think about like now if we go do some hot or exercise in a hot condition, like we could drink things like Gatorade, and they didn't have that. But basically, historians, anthropologists, even physiologists have kind of debunked this. So with the hot and arid environment in Africa, so they are right next to the ocean, there's inland rivers, there's trade routes. So salt was abundantly available in West Africa dating back years and years and years. The transatlantic slave trade, so obviously it's a terrible thing, not anything to like kind of downplay, but you have to think that a lot of the slave traders were treating these humans as like an economic commodity. So you don't want to get a shipload of people and then have them all die on the way there. So they still fed them, they still gave them water. It wasn't what we would consider adequate, but the goal was to keep as many people alive as possible. And then with New World slavery, there's not great medical records dating back to then, but when you look at like the major causes of death, like dysentery, it was one of the causes of death, and it did cause a lot of problems, but the major causes of death were things like pneumonia and tuberculosis. So things that you would expect if you take a group of people from one continent and put them on another continent without modern medicine. So it doesn't seem that this theory really holds up, and then there's actual data to refute it as well. So this is data from Curtis Morris's group at UCSF. So this is a group that in the late 90s, early 2000s did a ton of work on salt sensitivity in African Americans specifically. So a lot of their work was kind of phenotyping salt sensitive and salt resistant Black Americans. So most of their cohorts were all Black, and they did have a couple studies where they did comparisons. But so what we can see from this study is in the kind of black circles here, that's the salt sensitive cohort. And then in the triangles, that's salt resistant. And so if anything, if we look at the cumulative sodium balance in these participants, the salt resistant group had a higher cumulative sodium balance. And then if we look at percent body weight change, so if you consume more sodium and you're maintaining more or retaining more sodium and drinking more water, you're going to have a change in body weight. So that's just kind of a crude index of sodium retention. And again, if anything, the salt resistant group tended to have a larger increase in body weight. And then when we look at some of the other factors that might contribute here, so as we would expect, the change in mean arterial pressure is higher in salt sensitive patients. But then when we look at some of the underlying factors, there's a greater reduction in renal blood flow. There's a greater change in GFR divided by ERPF. And then with the renal vascular resistance, that's also elevated in the salt sensitive cohort. So it seems like what's really causing the salt sensitivity is renal dysfunction and a lack of a reduction in peripheral or a lack of peripheral vasodilation to kind of counteract that increase in water and cardiac output. Some of the things that may contribute to salt sensitivity, particularly in individuals with African ancestry. So there are some studies that I've just looked at urbanization. So this is not West Africa, it's more East Africa. But if you look at migration from the Luau region, which is more rural, to Nairobi in Kenya, which is more urban, you can see in this group of people who did that migration, there's a significant increase in both systolic and diastolic blood pressure. And some of the major things that we do see are a change in body weight, and then we see a significant increase in the urinary sodium to potassium ratio. So that would seem to be kind of more of a contributor to the increase in blood pressure as opposed to any sort of genetic selection. And then another reason why I'm very skeptical of the kind of genetic component to this is if we look at populations that have a high proportion of West African ancestry. So if we look at American Black individuals, pretty high percentage of West African ancestry, especially in the Southeast United States. But then if we look at, for example, Nigeria, so in West Africa or Jamaica, the hypertension prevalence is much lower. So that would suggest us more kind of a social component here, or a socio-environmental component. And so we know from AHA data that's updated every year, so consistently across time. So in blue, we're looking at the late 90s to kind of early 2000s, 2005 to 2010 in red, and then more recent data, 2011 and 2016. Across all these time points, we've seen that irrespective of biological sex, there seems to be that African Americans have the highest blood pressure when you compare them to both white Americans and then Mexican Americans is how they classified that here for the AHA. So one of the things that we do know is that hypertension alone is a predictor of salt sensitivity. So maybe these kind of baseline differences in resting blood pressure could be one of the things that's contributing. And as I said, so we know that there's this kind of population of individuals in the United States that does have a high proportion of West African ancestry. So we know that to be true. But it's pretty interesting, a recent secondary analysis of the SPRINT trial. So that's a large hypertension trial that actually kind of helped inform the 2017 guidelines. What they did is looked at West African ancestry. So we're looking at that on the x-axis, and then they looked at the hazard ratio of primary composite outcomes. So similar outcomes that I mentioned earlier, things like myocardial infarction, cardiac arrest, stroke, in the Black participants who were in SPRINT specifically. So if you self-reported as Black and then they genotyped and looked at West African ancestry, if anything, there was an inverse relation between West African ancestry and major cardiovascular events. So that would suggest that West African ancestry alone is not driving like cardiovascular disease. And one of the things that they mentioned there was that in studies to date, genetic variants only explain about 3% of hypertension. So I thought that was also interesting. So some of the things that we're trying to capture in our current study include social determinants of health and then health behaviors. So coming from an exercise physiology background, I'm interested in things like fitness and physical activity. And then with more recent collaborations, and just as I've been trying to read more in preparation for the studies that I'm doing now, I've been more and more interested in social determinants. So this is always a book that I plug during this presentation. It's called The Color of Law by Richard Rothstein, who's a lawyer and historian from Yale. And so we know that there was slavery, we know that there was Jim Crow, and then also on top of that, there's like a history of redlining in the United States. So basically with that, they would take neighborhoods or areas of like a city, and if there's a high proportion of Black individuals, they would redline that. So you could see that here in the book cover, where they've kind of drawn this border where there's this redlined district or area. And so that would kind of signal like, okay, we're not going to invest in this area. We're not going to bring in banks. We're not going to give housing loans. We're not going to invest in the schools. And then in housing covenants in other neighborhoods that were outside of that redlined area, they would literally have language where like, we aren't going to let Black or brown people live here. So that created these major gaps that we still see today in home ownership and in median household income. So they've just more recent data here that's plotted is median household income comparing majority white communities in the blue and then majority Black communities in the purple. And then if we look at median home ownership rates, kind of same thing, majority white communities compared to majority Black communities. So we do see a lot of socioeconomic disparities that could contribute to some of these things like salt sensitivity and hypertension. So that's one of the things that I'm trying to work on now with Thomas Fuller Rao. He's in human development and family studies at Auburn, and he had a large college sample, and his student Olivia kind of ran this study. So they looked at things like parental education, parental income, and then neighborhood disadvantage index. So they captured college students' zip code from age 0 to 5, 6 to 12, and then 13 to 18 years old, and tried to see if these different time periods might correspond more to things like blood pressure. And so we're interested in bringing these students back into the lab, and we're capturing diet logs, physical activity, and sleep. And that's sleep is another thing that they're interested in, but we're looking at vascular function, blood pressure responses to physical stress, blood biomarkers, and kidney injury markers. And so some of the data that we have gathered already are differences in both sleep continuity and duration. So there are large cohort studies, like I mentioned MESA earlier. So they found that African Americans tend to sleep on average about 40 minutes less than other racial or ethnic groups in that study. And we found that even in college students, that there's about a 30-minute discrepancy in sleep duration. And then if we look at sleep efficiency, which is a measure of like sleep continuity, like how often you're waking during the night, we've seen that there's a significant difference there as well. And then we're also interested in diet. Like I mentioned, we do habitual diet logs as well. So from larger data sets like NHANES, we've seen that there are differences in fruits and vegetable intake. That's largely mediated by socioeconomic factors. We know that if you live in like a food desert or a food swamp, you're more likely to consume energy dense foods that are kind of lacking in micronutrients and fiber. And then one of the misconceptions I've come across like in peer review and even just talking to people is that there's racial differences in actual sodium intake. And most studies don't show that. Under habitual conditions, like across most races and ethnicities, it's pretty constant. But one of the things that is different is potassium. So in that first bullet point, as I mentioned, there are disparities in fruits and vegetable intake. And so one of the interesting things that Curtis Morris, that same group, has looked at is changes in mean arterial pressure. And so in several studies, they've shown this racial difference when they compare Black versus white cohorts. But then in a couple of the studies that they've conducted where they've added potassium into the high salt diet, that seems to attenuate the racial disparity in salt sensitivity. So in terms of resting blood pressure and how that might contribute to some of the changes, we've even had some of our pilot data here. So we've looked at things like central blood pressure using the Sphygmocor Excel and seen a significant difference in central systolic pressure. And this is cohort on average in their early 20s. So again, mostly college students who are kind of on the tail end of their college career. A trend for a significant difference in central diastolic pressure, and then significant difference in augmentation index, which might kind of contribute to that change or the difference in central systolic pressure. And then we've also seen differences in ambulatory blood pressure monitoring, so nocturnal dipping. Specifically with diastolic BP, we've seen significant differences in nighttime blood pressure. And then when we look at the change, so here we're looking at delta systolic BP, so the change in blood pressure from the daytime to the nighttime, we've seen a significant difference in white compared to Black adults, and the same thing with diastolic pressure. So lastly, I'll just wrap up with some of the future directions, some of the work that we have in progress now. So salt and sympathetic outflow or sympathetic regulation. So Dr. Farquhar collaborates with Sean Stalker, who's currently at the University of Pittsburgh. He does a lot of preclinical work with sympathetic regulation, dietary sodium, and obesity. And so he has some data from the mid-2000s where he's done intracerebral injections of sodium chloride and compared that to controlled conditions. And so what we can see is that in response to glutamate, which is sympathoexcitatory, in the context of having a higher sodium chloride centrally, that there's this increase in both mean arterial pressure and then renal sympathetic nerve activity and splanchnic sympathetic nerve activity. And then they also had a not super recent, but more recent study where they looked at a kind of a mimetic of the exercise pressor reflex in rodents. So they can stimulate the tibial nerve to activate the triceps surae muscle, and they have a tension transducer here. So that kind of their model of in situ mimicking the exercise pressor reflex. And so even in salt resistant rodents, they found that under the high sodium condition, there's a larger increase in mean arterial pressure. So our goal while I was a postdoc was to try to translate some of these findings to human participants. So we did a controlled feeding study, a little bit different than the prior controlled feeding study that I mentioned. So these were 10-day diets, random order, and then our lower sodium diet was a little closer to what people might actually consume. So it was 1,000 versus 500 milligrams. But on day 9, we'd have people come in with the 24-hour urine collection, or start the 24-hour urine collection so that we could confirm that individuals were consuming more sodium. And so during this study, we're interested in measuring blood pressure responses to hand grip exercise or exercise pressor reflex activation. So there's a 10-minute overall baseline after participants were instrumented. Then we did another additional 2-minute baseline, 2 minutes of hand grip exercise at 40% of their maximal contraction, and then a post-exercise ischemia period. So you can see here we're assessing sympathetic nerve activity, so postganglionic muscle sympathetic nerve activity. And then we also have a pressure occlusion cuff, which you can see on the upper arm there. So we would have them squeeze for 2 minutes, and then shortly before they stop squeezing, we inflate that cuff, and that traps all the metabolites in their forearms. So that's activating the metabol reflex. So again, muscle sympathetic nerve activity and beat-to-beat blood pressure are our two outcomes here. And then this is kind of what we would see on our end. So this would be MSNA at rest, muscle sympathetic nerve activity just during that resting 10-minute baseline. As Dr. Lindbergh can attest, in healthy young females, sometimes you don't see that high of a resting muscle sympathetic nerve activity. And then during exercise, you can see a large increase in MSNA. So for us, we're interested in looking at the difference between MSNA during the hand grip or during the post-exercise ischemia and subtracting the resting so that we could calculate sympathetic reactivity. And so with that study, we did find that there's a significant interaction for time point times sodium. So it seemed that there was a larger increase in sympathetic reactivity with the recommended sodium and high sodium compared to low sodium. And we can see kind of almost a step fashion increase there when we just look at the change in burst frequency, comparing minute 2 of hand grip to baseline. And then when we look at the blood pressure responses, not quite a significant difference with the systolic blood pressure, but we did see a significant or close to significant difference in mean arterial pressure and diastolic pressure. So this data is kind of ongoing. It was kind of interrupted by COVID because the study's still going after I left. But that's something that we're working on to build the sample size of this study and kind of keep this study going. Just because we're kind of close with some of these p-values, so it seems like we're kind of underpowered here. But then one of the things that we did do, just because we noticed that kind of reduction in low compared to both recommended and high, we looked at hand grip, absolute force, so it's similar across all the diets. We looked at RPE, maybe that was driving some of the changes because we know that pain can contribute to blood pressure. That was also similar. And then we looked at blood lactate levels, which were also similar. So that kind of also kind of compelled us to keep that data collection going and seeing, what's really going on there. So future directions, going to keep our soup study going, hopefully get a small pilot study going here soon with another salt capsule study model, and hopefully future direction would be another controlled feeding study. And then one of the things that we're kind of moving on to is trying to see what we can do to prevent some of the negative effects of sodium. So we know there's all this data about blood pressure and blood pressure independent effects of sodium. We know that most Americans are over consuming sodium. So what are some of the strategies that we might be able to do to mitigate that? So this is my postdoc lab here on the left, where I got a lot of this research started, my current lab on the right. So just want to give a huge thank you to them because it wouldn't be possible without them. And thank you all for your time and acknowledge our funding mechanisms.
Questions when they're, so that they can get well on Zoom.
Oh, sure.
I'll start. Very nice presentation. My first question is, have you or anybody else in the field determined if salt sensitivity is related to increases in sympathetic nerve activity? And the second question is, have you determined if the salt intake increases sympathetic transduction?
Yeah, so the question is, has anyone determined if salt sensitivity is related to increases in sympathetic nerve activity and then also changes in sympathetic transduction? So there were prior studies where they found that sympathetic nerve activity, if anything, was reduced when you had a higher sodium diet. The problem was a lot of those studies used, again, like 500 milligrams for low sodium. So they may have been kind of volume depleted. And so it wasn't clear whether super low sodium was causing an increase in sympathetic activity or really high sodium was causing a reduction. So in the more recent controlled feeding study where we gave 1,000, 2,300, and closer to 7,000, we saw no difference in 1,000 versus 2,300, but we did see that sympathetic nerve activity was reduced with the high sodium condition. So maybe that's kind of barrel reflex, kind of counter-regulatory to an increase in plasma volume. In terms of sympathetic transduction, our group found that compared to even 1,000, 2,300, resulted in an increase in sympathetic transduction. And then more recently, this is something Dane and I were talking about earlier. Different groups look at transduction different ways, but Shifu's lab found that high sodium resulted in a greater transduction compared to low sodium.
I have a question about that pie graph. So you had those two pie charts on the bottom where you saw a greater increase in 5 millimole in the African American group. So is that poor sodium clearance? So if you're infusing it with saline, is everybody else just clearing it?
Yeah. So
Or is that-
yeah. Oh, sorry.
No, I just didn't understand the graph.
Okay. So this question is related to changes in serum sodium in the pie graph. So it seemed that the African American cohort had a higher percentage had the large change in serum sodium. And so the question is, I guess, are they having a harder time clearing or buffering? I think based on some older data in the I think it was the late 70s, there were some inpatient studies where they compared kind of all male again, but it was black versus white males. They did some furosemide studies and hypertonic saline infusion. And so what they typically found was that over the course of 12 hours, there were larger changes in serum sodium in the black male cohort compared to the white male cohort. And that would tend to normalize over 24 hours. So maybe at least acutely, they do have a harder time buffering the changes in serum sodium. And maybe part of that could be due to, there are kind of well-documented racial differences in the renin angiotensin, aldosterone system hormones, at least in the circulation. So it seemed like they probably do have a hard time buffering that, at least acutely.
Is there any differences in regards to the reduced endothelial function with added sugar and sugar within whole foods? I mean, we know processed is just horrific, but just the salt load as opposed to cheese, for example, or something that's a high sodium food. But because with obesity, you don't necessarily see reduced endothelial function. They likely overconsume sodium though, but I don't know if you.
So is the
So
question
I
kind of
guess
like the
added-
interaction between sodium and sugar?
No, I'm sorry. Added salt or salt naturally occurring in foods.
So salt from
So,
more processed foods?
Yeah. Yes.
Yeah, so the source of salt, and there are some data suggesting that it does matter, more or less the background diet. Does the background diet matter? And so there are the dietary approaches to stop hypertension, the DASH sodium trial. So for that trial, they looked at low, kind of more medium and high sodium. But then the main difference was DASH diet versus standard American diet. So the DASH diet is higher in fruits and vegetables, like low fat dairy, whole grains. And so what you do see is that there's less of an increase in blood pressure in the context of a DASH diet, but there's still an increase. So you have lower blood pressure on the low sodium DASH diet compared to the high sodium DASH diet, but compared to the standard American diet, DASH diet does seem to be protective. So.
One more question. Have you considered examining if increased potassium intake could correct the negative effects of sodium intake on vascular?
Yeah, so the question is, does increased potassium intake counteract the negative effects of sodium on the vasculature? So that's sort of been done. The reason I say sort of, so Shannon Lennon at Delaware has a study, it's actually an R01 now, focused on potassium. So it's not bananas, that's the thing. So it's framed as a potassium study, but they actually increase just fruits and vegetable intake. So then it's not really a potassium study. It's more it could be fiber, magnesium, dietary nitrate. So I don't know of anyone who's specifically given a source of just potassium and determine if that prevents endothelial function, but her data where they do give more fruits and vegetables, that does seem to prevent the negative effects of sodium on the endothelium.
In any of your studies, have you looked at the ratio of sodium to potassium excretion? Is there a difference in people who are sodium sensitive to sodium resistant, the amount of potassium they retain?
So are there any differences in the sodium to potassium ratio in salt sensitive versus salt resistant individuals? It may have been one of those things where I just looked at it like one random afternoon, but off the top of my head, I can't remember.